Hello everyone, good afternoon and welcome to join our webinar today. I'm Cassie, the host for today's webinar. On behalf of GenScript ProBio, I would like to thank all of you for taking your time to attend today's webinar. So a little background for those who are new to us, GenScript ProBio is the subsidiary of GenScript Biotech Corporation, proactively providing end-to-end CDMO service from the drug discovery to commercialization with proactive strategies, professional solutions, and efficient processes in cell and gene therapy, vaccines, biologics discovery, and antibody protein drug to accelerate drug development for our customers. So in today's webinar, we will include 2 presentations, and each presentation will take around 30 minutes. We will attend to the Q&A session after we finish both of the presentations. You can submit your questions at any time point during the webinar using the Ask Questions panel situated at the menu on the right. So now I'm delighted to introduce our first speaker for today, Dr. Brad Walsh, the CEO of Glyterix. Dr. Walsh founded Glyterix to bring to market an innovative antibody for treatment of solid tumors from cancers. He began his career in biotechnology with a PhD in protein chemistry, and he has led research groups in government agencies, universities, and hospitals, and he was part of the key of establishment of a National Proteomics Facility, APAH. He continues to be active in research collaborations and academic supervision, and in 2015 was a joint recipient of one of Australia's most prestigious science awards, a Eureka Prize for interdisciplinary research. He has co-authored 105 peer-reviewed papers and book chapters and had 7 patents under his name. So in today's webinar, Dr. Walsh will be discussing about development of an exciting new radioimmunotherapy for cancer patients. So now let's welcome Dr. Walsh to present. And Dr. Walsh, over to you right now.
I'm the CEO of Glyterix Limited, and I would like to thank the GenScript ProBio team for inviting me to present to you about something we're very excited about, which is a new radioimmunotherapy for cancer patients that has been developed by us. So, forward-looking statement from the company, and let me tell you about the target, which is Glypican-1 or GPC1. We think it's a great cancer target. It has clinical novelty and already proven safety. Expression is seen in 8 solid tumour types, most of which have high unmet need. And it plays a critical role in invasion and metastasis, so it's a great target for aggressive tumour types. High GPC1 expression is often associated too with poor survival and poor clinical outcomes. We now have 4 families of patents and 70 registered patents from those 4 families, and we have a clinical program which is about to enter into phase 1B. So, brief overview of the company. We call ourselves a theranostic company, and what we say about that is image the cancer and treat the cancer. The addressable market for those indications could be up to $7 billion US. We target that Glypican-1 with an antibody. We have a first-in-class molecule called miltuximab. It has a high degree of specificity, and we have seen no off-target binding. The protein itself is cell surface in prostate, bladder, pancreatic, esophageal, ovarian, breast, glioblastoma, lung, and most recently colorectal tumours, and is not detectable in normal tissue. So far, we've been able to complete a phase 1A safety study with no drug-related adverse events. Because it's shown such a strong safety study, we're progressing to the next stage, and that's what I'll tell you about today. We have a manufacturing consortium led by Glyterix and funded by the Australian government. We've now completed the cell banks and manufacturing of chelated antibody, so we can add to it the radioisotope we need for the radioimmunotherapy. So next steps are manufacturing GMP material and executing on our phase 1b study. As I mentioned before, we have a good IP portfolio and a growing number of peer-reviewed publications demonstrating that the science is robust. So the molecule itself, Glypican-1, is expressed in those solid tumours, as I said, associated with poor prognosis, and also resistance to standard of care treatments. Our antibody already we've seen in the clinic and we've seen evidence of targeting. So let me talk a little bit about theranostics. Theranostics is a growing field and there are a number of companies that are doing really well in this field. And what it talks about is combining therapy and diagnostics together. So our base molecule called miltuximab is the anti-GPC1 antibody. And we can label this using a chelator with either zirconium-89 for imaging or lutetium-177 for therapy. So a patient can be administered a small dose of antibody with zirconium-89. This can then be localized using a PET-CT camera to not only show that the tumours are there, but also to show that there is a possibility this patient may respond to the therapy which they can move on to, which has a stronger beta emitter called lutetium-177. So GPC1 is critically involved in cell growth, invasion and metastasis. It's a co-receptor to quite a number of growth factor signaling molecules. We've shown that knockdown inhibits growth, proliferation and migration. Knockdown in vivo inhibits primary tumour growth. So, for example, in a prostate cancer subcutaneous model, and also metastasis, in this case, with a PC3 intracardiac metastatic model. Expression is restricted, so there's no reactivity of the murine parent antibody with 20 normal cadaveric eye organs. And preclinical safety studies demonstrated safety of dosing up to 50 milligrams per kilogram in mice. So here's a little cartoon that shows you the GPC1 molecule shown here in yellow, and it is a proteoglycan, so it is glycosylated with a peptide backbone. It acts as a co-receptor for signaling with molecules like TGF-beta, FGF, frizzled, and patched. So as a co-receptor, signaling through the TGF-R1 and R2 receptors, it can promote epithelial to mesenchymal transition and invasion in tumour cells. The TGF-beta itself can activate CAFs, which influence the tumour as well as the tumour microenvironment. Glypican-1 sequesters FGF in proximity to the FGF receptor to allow it to drive signalling via an FGF pathway, which, as you can see here, can increase things like angiogenesis, migration, invasion. Delivering Wnt to frizzled, GPC1 can influence beta-catenin signalling, which drives both metastasis and invasion. And finally, hedgehog. This signaling is influenced by GPC1 and promotes the activation of TME cells, such as stroma cells. So you can see quite an important molecule. And here, as I mentioned, again, a good study here by Cato et al. And this was on fresh frozen normal tissue from 3 donors. And you can see here very clean staining, no antibody staining, in contrast to the last panel here, which shows some tumour staining. So, as we said, no demonstrable reactivity with normal tissue. In contrast to this, work we did with the murine parent antibody called MIL38. So this is the miltuximab fully murine monoclonal. You can see here in normal prostate or in an secondary antibody control, no staining. With prostate cancer or prostate cancer metastases, very clear staining. Down here, a xenograft from DU145, which is quite an aggressive phenotype prostate cancer cell line. And here, secondary antibody control with a bladder cancer tumour versus staining with the bladder cancer tumour using the MIL38 antibody. So very strong, clear staining in tumour tissue, but not in normal prostate tissue in this instance. Again, here is a study on a mouse cross-reactive anti-GPC1 antibody. This is from a group in Japan, Harada et al. And you can see here, anti-GPC1 antibody that recognises mouse GPC1 was dosed at 50 milligrams per kilogram for a toxicology study, and there were no effects on either serum chemistry or blood cell counts that were attributed to that antibody. So you can see non-significant p-values in comparing the controls and PBS with the anti-GPC1 monoclonal antibody. Lastly, again on this cross-reactive anti-GPC1 antibody, no histologic changes observed in many key organs. These are H&E staining of paraffin-embedded tissue after 7 days of treatment with the antibody. And you can see quite clearly between the anti-GPC1 and various controls in both male and female mice, there is no difference. So very good safety profile that led us to move into patients. So this is not only a first-in-human study, but a first-in-the-world study that was demonstrating key outcome of safety and tolerability with a secondary endpoint of dosimetry and targeting. So in this study, there were 12 metastatic prostate cancer patients. They had to have between 2 and 15 metastases and had to have stable disease. If you're interested in reading more about this, Sabanathan et al. Is our publication on this, which came out in 2021. So as I said, primary endpoint, safety, tolerability. So the primary endpoint was met in all patients. Single dose of antibody up to 25 milligrams, so between 0.01 to 0.33 milligrams per kilogram as we increased the dose across 4 cohorts. And we saw no drug-related adverse events. The secondary endpoint, dosimetry and targeting. Targeting was present in more advanced metastases, and I show some here. The best time point for imaging was 48 hours post-infusion, and dosing at 24 milligrams increased blood half-life, so we were able to keep the antibody in the circulation to allow it to accumulate at the tumour site. Importantly, we also did dosimetry, showed that the amount dosed was well below safety limits. We're also able to estimate the kinds of amounts we would need for higher energy isotopes such as zirconium and lutetium. I should have said that what we were using here was actually a gallium isotope, and these images come from a gamma camera. So you can see here in patient 2 who had metastases in his bone, very nice targeting of the miltuximab, and in this patient who had mets, cranial mets, so these were in the skull. Again, these cranial mets were quite evident in the uptake at 48 hours. What is here is a control pot of the gallium that is used for correcting the imaging and the dosimetry. As I mentioned, here is the patients from 1 to 12. Adverse events were graded from 1 being lowest and 4 being highest. And you can see there were both hematologic, renal, hepatic, cardiac and clinical adverse events recorded. And in every case, the patients did not see drug-related adverse events to the drug. So because of this great outcome, we have moved to the next phase, and that is to go from the first in human and moving into clinical phase 1B study with further studies planned throughout the next 7 or 8 years to take us through to market. The indications are given here: prostate, bladder, pancreas, glioblastoma, lung, ovary, breast, esophagus. These first three were in our phase 1A basket study, the safety study, so they've been through here, and we're taking them into phase 1B. Because of the data that has been published already, we're actually able to accelerate lung, ovary, and breast into that 1B study. We are currently raising some funds to support that. Additionally, we plan to do a first-in-human with glioblastoma with our partners at the University of Queensland, and I'll talk a little bit more about that later. So we've now completed the manufacturing required to chelate zirconium-89 to miltuximab. To chelate that, we use a different chelator. We use DFO rather than DOTA. And so you can see here, this is glioblastoma, or in this case, prostate xenograft on the flank of the mice. These mice have been dosed up with the zirconium miltuximab, and this is a PET CT scan. So the next step for this is delivery to patients, so that we can see where the tumour is located in the patients and who is suitable for therapy with miltuximab containing a stronger beta emitter. And that stronger beta emitter is lutetium-177. This is another publication of ours by Yet et al. That was published in 2020. And you can see here a couple of experiments. So these two here show antibody with only the DOTA cage on it, so no radioactivity on there. And then two different levels of radioactivity, 3 megabecquerels and 10 megabecquerels. You can see here that with DOTA and the mice were given a prostate cancer tumour on the flank. That was allowed to grow to day 24, and then treatment was commenced with a single dose of antibody. You can see if there was no radiation on there, the tumours continued to grow until the termination of the experiment. You can see if it was 3 megabecquerels, there was an inhibition of tumour growth, and with 10 megabecquerels, not only an inhibition, but in fact a decrease in tumour growth. The Kaplan-Meier survival curves are shown here, and you can see a good increase in survival, particularly, of course, with the 10 megabecquerel. In another experiment given here, this is a 6 megabecquerel dose, just to show in the first the tumours, in the second the Cherenkov radiation, and then an overlay. And of course, what you can see here is that if the animal only had the DOTA with no radiation, no accumulation, but here at day 3 and day 5, increased accumulation, very clean, only on the tumour itself, so we can't see any targeting elsewhere in the body when we get through to day 5 and 6. So the miltuximab showed specific targeting and retention in the tumour, and this is what we need for radioimmunotherapy. We need the antibody to target and to stay there while the radiation kills the cells. A very good safety profile, so the safety data from these mice showed no adverse effects on other organs. Dose-dependent inhibition of tumour growth, so as we went to 10 megabecquerels, very good effect on tumour growth and improved animal survival. So for a little while now, we have had a federal Australian government grant called a CRCP, and that has allowed us to put together a manufacturing group as well as a study group to build batches of clinical grade product to put in trial in the clinic. So around GMP manufacturing, importantly Cytiva, as well as the CSIRO and University of Technology in Sydney. On the radiation side, OzPEP and ANSTO, and then finally Macquarie Health and Macquarie Medical Imaging with our principal investigator, Professor Howard Gurney. So it became clear to us as we began to do that we needed some additional capability, and you can see here some pipeline development. So, for example, in the glioblastoma study we've been doing with the Centre for Innovation in Biomedical Imaging and Technology, which is an ARC hub, and that's around glioblastoma, but we have really needed to access some expertise and some people who not only are able to produce these things but understand the timelines of a startup biotech company. So we've entered into collaborations with, importantly, GenScript ProBio in cell line development, and also more recently with WuXi in chelation chemistry. So the reason that we were interested in GenScript ProBio, we have in fact made a antibody pair for ELISA before on GPC1, and GenScript were able to help us with that. So we went back to them to talk about whether we could get a high yielding clinical cell batch, clinical grade cell bank that we could use. So we looked at the GS system and the antibody expression levels that we might expect from this, and we started a program with them through this year to work with the team in GenScript ProBio and the team at Glytherix. So this took us 7 months, went through 3 stages: first stable cell line development, primary cell bank stability, and then, of course, we needed media screening and clone evaluation. The really fantastic outcome we got from this was the top 2 clones showed titers above 8 grams per litre. So we're really excited about this. This is one of the key things for a company like ours, who is an antibody company. We need to have a reliable, high-producing titer of cell bank for us to be able to supply antibody for our studies and, of course, in the future for our commercial product. So the plan, and we're partway through this now, of course, is the GMP monoclonal antibody production and the bioconjugation. So we're progressing through this stage at the moment. And parallel with that, working with ANSTO on the development of the radio labelling. So, of course, as you can imagine, we've already done some of this work in our phase 1A study, but we are now developing this further so that we can add to that the zirconium-89 or lutetium. And as we start to do that, we are now planning our protocol with our principal investigator for our phase 1 clinical study. Of course, during that time, we will need to make various doses for the patient study. The other thing that we've done is we've looked at actually accelerating the patient dosing a little bit, and we're currently reviewing that. We think that probably the timeline for that will be about 9 months now. So at the moment, the study looks like this, and this gets back to what I was saying before about theranostics. It's going to be a phase 1B basket trial at Macquarie University Hospital, where first patients will be screened with zirconium-89 miltuximab. So that's the imaging agent. They'll have PET-CT. As soon as we screen a cohort and we identify 3 patients, that cohort will be admitted to the first dose of lutetium miltuximab, which is the therapeutic. So we go from the diagnostic dose to the therapeutic dose. And you can see here that it is a staggered set of cohorts. So once cohort 1 are dosed and the drug is deemed to be safe, cohort 2 will begin at a higher level of radiation. And we're aiming to go up to about a 1.5 gigabecquerel dose of radiation in the patients. The patients can have up to 3 doses. They may not need that. They'll be observed, but they can have up to 3 doses. And part of the reason to do that is to dose fractionate to make sure that we don't have any adverse effects on bone marrow in these immunocompromised patients. I mentioned also that we're looking to start another study. This is zirconium-89 study at Royal Brisbane and Women's and the Wesley Hospital with a group of people that we've been working with for some time now at CIVIT, most importantly Professor Chris Threcht. This is to enroll some patients and have a look at infusion of miltuximab intravenously into those patients. Now, those of you who know something about glioblastoma and antibodies may say they won't cross the blood-brain barrier. However, we have found that in contrast positive patients with a compromised blood-brain barrier, full-size antibody is in fact able to cross over. So these patients will be later stage patients who have very little other treatment options. So we're very excited to do this. We think that it's perhaps something new in the fight against glioblastoma, which is an insidious disease. So miltuximab might be a great thing to do in this case. As I said before, it's really important in this area to also have patent protection. And you can see here we have 4 families of patents that have been through national phase now in many countries. It's nearly finished. PCT was completed some time ago. There's a list of countries down here which include China, the US, Japan, as well as Australia. So we're finalising that, so good patent estate. Importantly, our coverage goes for quite a few years yet, and we have just lodged a new provisional on a humanised antibody, which will be our 5th family, and that was lodged about 2 weeks ago. Importantly too, I think is peer-reviewed papers on this technology. And you can see here just a little snapshot of the peer-reviewed papers that we've published. So we have 16 peer-reviewed papers now. We have 1 more which is about to go under final review. So there's a very good body of work that shows this. Additionally, there have been some R&D groups who have been working on GPC1 in various places. And so there is a very good body of literature to suggest that GPC1 will be a very important target in solid tumours now and in the future. And because of this, we really think that there is a very good addressable market around the world, and this is a very wordy slide, but it just estimates what we might expect in terms of market share in a number of what we think are cancers with unmet need. So they're niches such as triple-negative breast cancer or checkpoint inhibitory refractory bladder cancer, glioblastoma, enzalutamide-resistant prostate cancer. And you can see here some estimates of the peak therapy and imaging and then the total sales from that. I'd like to thank, before I finish, in particular Dr. Douglas Campbell and Dr. Yanling Liu, who are head of R&D and head of CMC. Douglas and Yanling have really been driving this program in the laboratory and in the clinic, and have worked very closely with Wuxi and with GenScript ProBio and our other partners, and very important on the team. The other people who have been key to us, of course, are our clinical advisors, Professor David Gillett, who is Director of Medical Services at the Macquarie University Hospital, but also Dr. Neil Shaw, who is CMO of Surgery Urology at Genesis Healthcare, and a past president of the large urology group Practice Association. Dr. John Babbage, who is now president and CSO of Ratio Therapeutics, was formerly at Weill Cornell, and of course our PI, Professor Howard Gurney. So I would like to say thank you there, but I would also like to say that this is a really important area that is going to give cancer patients new alternatives, new therapies, so that when they fail standard of care, they will be able to go on to something else. So we're very strongly committed to bringing this to market. Thank you very much for your attention, and I look forward to your questions in a little while.
All right. Thank you, Dr. Walsh, for the excellent presentation. If you have any questions, please do remember to type it on the Ask Questions panel. And meanwhile, please also follow our LinkedIn account, GenScript ProBio, where you can view more online resources such as new service launch and other webinars as well. Thank you so much. So again, just a reminder, we will get to the Q&A session after we finish the next presentation by Mr. Jiang. So now, let me please introduce the next speaker, Mr. Ruby Jiang. He's the Director of Antibody Process Development and MSAT. He received his master's degree in biotechnology from Northeastern University in 2014, and at the same year, he is also the process engineer at Sanofi Genzyme Upstream Process Development of Antibodies and Recombinant Proteins. From 2014 to 2020, he worked in the Process Development Department of Merck Sharp & Dohme, responsible for the upstream process development, process characterization and validation, as well as leading the upstream process development of Merck Sharp & Dohme's continuous production and process online analysis PAD upstream development. During this period, he participated in the publication of 7 academic articles. From mid-2020 to 2022, he worked in Hangzhou Yi'an Jishi as Deputy Director and responsible for Upstream Process Development Department. And also, he's in charge of the CDMO Upstream Process Development and also promoted to enhance the fed batch process and continuous perfusion process platform. So in this webinar today, Mr. Jiang will discuss about R&D enabling CMC strategies for monoclonal antibody and bispecific antibody, which could be faster and also better for you. So now I will hand over the floor to Mr. Jiang now. Mr. Jiang, please.
Hi, everyone. Many thanks for Dr. Welsh's presentation and many thanks for Cassie's introduction. My name is Ruben. So today my topic is about R&D enabling CMC strategies for mAb and bispecific mAb. And overall, we want this process as a CMC people, we want to make the CMC process development and manufacturing faster and better. First is about a quick introduction about GenScript ProBio. So GenScript ProBio is a global company with customers from all around the world. In New Jersey, we have a manufacturing facility for plasmid and clinical manufacturing GMP facility. And also in New Jersey, we have a GenScript ProBio headquarters for North America. And for Europe, we have a EU headquarters in Amsterdam, and in China, Nanjing, which is where the GenScript ProBio's headquarters is located, and we have our biologics development, process development, and manufacturing facility in Nanjing. And in Zhenjiang, we're building our phase 3 clinical and commercial manufacturing facility for antibody production. And in Tokyo, we have a ProBio cell office, and in Singapore, we have a GenScript Asia Pacific headquarters. Overall, we are very fast-growing CDMO company. Today, I'm going to focus on the right side of the slides. You can see so far we delivered more than 110 cell line projects for IND filing, and we finished the 71 CMC and CMO projects. And all together, up to now, we got 19 global IND approvals, including the ones from China, FDA, and EMA. We altogether delivered 77 GMP batches and went through 43 customer audits. And the manufacturing we're conducting applies for FDA, EMA, and MDA GMP standard. Most of our project is antibodies and proteins. 46% of all CMC projects were non-mAb proteins, and they're more difficult to develop and more difficult to manufacture. So, for example, 19% of the projects are bispecific and tri-specific projects, and 22% of all CMC projects are recombinant proteins. On the right side, I'm showing some of the customers that got IND approval and some of the projects that are ongoing. And on the right bottom, you can see the 5 formats we have been worked before for now. We have scFv fused with antibody, and we did in vitro assembly for customers, and we worked with knopped in whole formats, and the mAb in cyclotin connection, and also scFv connected with scFv formats. For now, we have a 2,600 liter GMP manufacturing capacity that is in Nanjing site. And by the end of this year, actually, we are going to launch our fill and finish line that makes all the drug product and drug substance production in the same site. And by the end of next year, we will have another 16,000 liter manufacturing capability ready. And it will have 8 2,000 liter bioreactors and with fill finish on site. Since host cell line is the start of the CMC project, I want to talk more about the host cell line, because if you have a right clone or a perfect clone with high productivity, and will help the CMC projects a lot. In GenScript ProBio, we have our CHOK1 genes wild type cell lines that's licensed from ECACC. And also we developed ADCC plus cell line, which can produce the afucosylated antibody. And both cell lines are adapted to suspension, and they can grow in animal components-free culturing, and they can compatible with the chemical defined commercial media. In terms of the vector, we also have our in-house vector system, one called DGV system and another one called 1.0 up. Most of the time in our process development, we will do these 2 vectors in parallel and find the best vector and the host cell line for the client. In normal circumstances, the 1.0 up will have a higher titer for mAb and bispecific antibodies compared with DGV. And for protein projects, we have the 2G unique vector, which we license from Proteonic, which is more specifically for protein production use. In the same time, during the process development, we have our clone ranking and screening platform, which is fully in compliance with FDA, EMA, and NMPA's requirement, and they are high throughput. So in short time, we can screen more clones, which means we will have a higher titer clones. This is a quick summary about how is the productivity of GenScript CHOK1 GS cell line. For mAb projects, on average, we have 5.48 gram per liter. And the highest one so far we got is 9.86 gram per liter in fed batch process. Population cell density. And if you look at the picture on the left bottom, more than 50% of our project final titer was more than 5 gram per liter. And more than even 10% of our project got the final titer more than 7 gram per liter. And there was no any projects from this cell line that has a titer less than 3 gram per liter for monoclonal antibodies. If you look at the right side of the slides, the CHOK1 genes system was also used to express different types of molecules for monoclonal antibody, bispecific antibodies, and recombinant proteins. As I said, for mAb is 4.48 gram per liter on average. And for bispecific antibody, we have on average 3.47 gram per liter. You can see there's a larger variation for the bispecific antibody. It's more because bispecific antibody or trispecific antibody, they have many different formats. So because of the variation of the formats, their titer was also varies a lot. However, if the clients need high titer to reduce the production cost, we can always use the intensified fed batch. Using that intensified fed batch mode, we can double the titer without impacting the product quality attributes and without impacting the CMC progress. And for protein projects, we have on average 2.16 gram per liter. A little bit about ADCC cell line. The reason we develop ADCC enhanced cell line is because it's easiest way and is a way that you don't have to file patents or fight patents with other companies. By using this cell line, you can just having your product ADCC enhanced without worrying any lawsuit or any patent issue, because most of the additives that can increase your ADCC is already filed by some of the competitors in the world. Our ADCC enhanced cell line has a similar growth profile and productivity as the CHOK1 gens cell line. And the product from the ADCC enhanced cell line, you can see on the top of the graph, there's completely no G0F at all because the fucosylation was greatly reduced by using this cell line. And for a case study of the ADCC activity on the left, you can see the affinity to the specific target was increased by 3 to 4 times. And on the middle of the graph, you can see the ADCC activity was increased by 10-fold by afucosylate, the N-glycan modification. And on the right, you can see the EC50 was reduced by 6 times. In conclusion, using this cell line, the ADCC effect was significantly increased. Another common question asked by our clients was, I have 3 or 4 candidates, which one I should choose? And for those needs, we also have our developability assessment. We recommend to do the host cell and the vector system cell pool to produce these target candidates. By doing this, we can keep the product quality consistent between animal studies, developability studies, and CMC studies. Usually, we would pick 3 extreme conditions, for example, high temperature to assess the room storage stability, and we look at the low pH to look at if during the purification if we have any issue, and also we will look at the freeze and thaw to support the shipping and the storage. And by testing the quality profile, we can select the best candidate for following CMC development. In the table on the right, you can see we have a candidate A and B and C. And after all those tests, and after we look at all this purity profile, quality profile, you can see the candidate C would be the best one in practical for CMC development and manufacturing. Last but not least, I want to talk more about the CMC strategy for mAb and bispecific mAb, because we have been doing this for a while, and I want to share some of our experience with other people in this industry. Overall, we have mAb and bispecific mAb platform in ProBio, and for mAb, the key consideration a lot of times are faster timeline, and the clients want to be cost efficiency, and so we would recommend to do the fast CMC trial that's using the fed batch process And the timeline, it takes from DNA to tox only takes 6 months, because once you have the tox batch delivered, the clients can use this product, the produced drug substance for tox study, for animal studies. So the CMC process will no longer on the critical timeline after the tox batch is delivered. That's why I specifically list the time from DNA to tox. And for bispecific product, a lot of times the key consideration is whether we can remove the byproducts, and in the same time, we want to fully optimize the process, because sometimes bispecific antibody is more fragile, depending on the specific formats. And we would recommend either fed batch process or intensified fed batch. It's really depending on the titer needs of the final product. And from the DNA to tox, it takes one more month compared with the mAb, because bispecific antibody needs more downstream tweak compared with the mAb. So from DNA to tox, it takes 7 months. I'm showing a quick timeline for the mAb development. If we go with a fast CMC project, it will take 6 months from the DNA to tox. And as you can see, at the end of month 6, the tox batch will be delivered to the clients for the tox study and in the animals. And it is guaranteed that in the 200 liter batch, we will deliver more than 0.5 gram kilogram product. And in a 500 liter, we will deliver more than 1.2 kilogram product. For bispecific antibody CMC, there are several challenges. For example, low titer and low product stability, and challenges in the purified byproducts. And last but not least, there are also a lot of analytical methods development needs, because sometimes the product and the byproduct are so similar to each other, it's hard to distinguish. So analytical department always have to work harder in order to prepare the specific bioassays to distinguish the byproduct and the target product. For low titer, we always modulate relatively expression of the polypeptides by optimizing the plasmid ratio to reduce mismatch, and in the same time improve the titer. And also in upstream and CLD, we have a high throughput screening that can screen the best clone, and also screen different media combination. In upstream, we do the process parameter optimization, and we can do intensified fed batch without impacting the product quality attributes. For purification, a lot of times we have to remove the aggregation and mismatch, so we utilize the multi-model resins based on the different types of the impurities, and also you have to tweak different process parameter in order to optimize the purification process. For formulation, is similar to the mAb. You have to evaluate the effects of pH and the buffer system and concentration to the formulation. And we have a lyophilization development capability. So if this product is truly not stable, we can always use a lyophilization process to dry freeze the product, which will have longer shelf life in the future. For analytical development, we would focus on the detection of the homodimers because those are the byproduct that's sometimes hard to remove by downstream. In the same time, we are capable of designing the binding or functional bioassay based on the mechanism of the action. Cell line development, like I just introduced, we would copy the line in different vectors. So by adjusting the vector ratio, we can have a higher titer in the same time, have less aggregation from the clone. And on the right, I'm showing that with Fc region containing bispecific antibody, our titer is on average 3.45 gram per liter. And some of the product is lower than that, and sometimes the product is higher than that. It's more product specific, but on average, the titer is 3.45. And for cell culture process development, it is similar to mAb. There's not too much difference. We have to optimize all the parameters, including temperature, pH, agitation, PCO2, osmolarity, and culture duration. For the culture mode, we have a fed batch process, intensified fed batch process, and perfusion. Most of the time for mAb, fed batch process is good enough to reach a 5 gram per liter titer. And for intensified fed batch, we can use for bispecific antibodies because the titer, if the clients have a high requirement for the titer, we can use intensified fed batch. And if this product, some of the product are not stable in cell culture environment for a long time, so perfusion, we would use perfusion process to address those products. These are some of the cases that we have titer improved in different ways. For example, amino acid supplementation, and on the right you can see pH adjustment also have a quite impact to the productivity. And on the left, on the bottom left, you see that high density inoculation, which is a fed batch process, can really improve the titer by sometimes more than 100%. And also temperature shift is also something that we will test during our process development for upstream. For product quality consideration on the left, you can see some of the product, they're not stable in fed batch cell culture process. So we use perfusion process to exchange those products out after they expressed in the expressed out of the cell, they are exchanged out of the bioreactor and keep the cool. In that case, they can keep stable. And on the right, you can see as the cell culture duration goes longer, we have aggregation come up as the impurities. So in this case, during the process development, we may have to consider the benefit and the concern by prolong the culture duration. And also, we have different ways to improve the glycosylation and cellulation modification. We have to add different supplements, different additives, small molecule additives most of the time. And on the right, if we want to do biosimilar, a lot of times the culture media screening is involved. We will screen different large amount of media to select the best match for the target product. For purification process development, the focus is always byproduct removal. And for aggregation, we have different columns to address those. In the same time, there are fragment and mismatch. We use the affinity column, ion exchange column, and multi-model column to address these issues. In this slide, I'm showing a good example of using the multi-model resins. On the left column, you can see the recovery rate. For the traditional cation exchange, the recovery rate is only 15%. And for using the multi-model resin, the recovery rate increased to 70%. And for C column, using the CHT is also a multi-model column. The recovery rate is increased to 75.9%. And the multi-model column have increased the recovery rate, but in the same time, it also have a higher purity. You can look at the SEC profile that's both the resin B and resin C have a higher SEC level, the mAb pattern. For homodimers removing, still on the right graph, you can see for anion exchange column, the homodimer after purification, it still has 38% of the homodimers, and for cation multi-model column, it only have 0.8%, and the product purity on the right for the traditional cation exchange column is only 61.4%, and for the multi-model resin is reached 99%. For formulation development is more similar to the monoclonal antibody product because still we went through those accelerated conditions to test the rate of aggregation in liquid formulation. And also we have the accelerated degradation that we can find at low pH. So we can determine the formulation based on the aggregation and degradation data. And for analytical method development is always another big part during the CMC development. And each, depending on the impurities, we have analytical group will have to develop a different assay to detect the specific impurities. For example, if the ICIEF is different between the product and the byproduct, we will use ICIEF to distinguish the difference. And if there's the target product and the byproduct have different molecule weight, we will use SDS page. And we can always use LCMS to monitor the undesired pairs, and also the monitoring the process development, also monitoring the manufacture of the final product. For bispecific antibodies, because it has two targets compared with monoclonal antibodies only have one, so it is more tricky to develop the bioassays for two targets. For this one, our bioassay group developed different strategies to verify the mechanism of the action for different target product. If we have two products that is dual immunomodulars, then we will develop two different cell-based assay to separately assess the potency of each antigen target arm of the bispecific antibody. However, if we have the T-cell engager, then we would do a binding ELISA to assess the binding activity of the both antigen, and we will develop one cell-based assay to assess the T-cell activation. As you can see on the following graph is a good example of this strategy. And ProBio has a well-established bioassay platform that can accurately measure the target MOA for various bispecific antibodies. We have cell-bridging bispecific antibodies, and for dual target blockade, we also have a corresponding cell lines. And for dual immodulars, we have a corresponding cell lines and a target. Because we have our own cell engineering group, this work can be done very quickly. I'm going to provide a quick case for a product. This product is already filed in IND for FDA and NMPA. During the cell line and the upstream process development, we did media screening and amino acid supplement to improve the titer by 62%. And again, we add manganese to lower the MEN5. For purity improve, we tweaked the different process parameters to increase the recovery rate and purity from 84% to 98% for the purity profile. And for bioassay, we selected a suitable bioassay to develop the assay to detect the potency of the multiple targets. This is a case one. So by media screening, we improved the titer by around 40%. And after optimize all the parameters, the final titer was 1.6 times higher compared with the original titer. And because we also, our clients may have MEN5 problem because MEN5 is more than 10%. So we add manganese to reduce the MEN5 to under 10 level. And so for purification, we used different resin and tweaked the different specific parameters. At the end, both the affinity column and the CX column have improved greatly. For bioassay development, because this is a tri-specific antibody, it is harder. What's the final strategy was we have two bioassay. The first cell assay is targeting the target 1 and target 3, and the second bioassay was targeting target 2 and target 3. By doing this, we can verify both the mechanism of this tri-specific antibody. The case study 2 is an in vitro sample of the bispecific antibody. For this one, we have the anti-target 1 connected with the linker first, and the second part of the scFv was connected to the linker. And in the third step, both scFv part was connected together and the linker was removed. In this step, again, we optimize the linker conjugation for scFv1 and 2 in the first place, and after all, we optimize the final conjugation step. So the ratio of the recovery was improved from 29% to 64%. That's probably near the end of my presentation. So in conclusion, BroBio has a leading bispecific antibody CMC platform, which can break through many CMC barriers. We have a high expression system. We have a good yield. In the same time, we have a strong analytical assay platform. And our CHOK1 genes and the CHOK1 ADCC system can provide the clients world-class productivity and also ADCC enhancement. And ProBio has a well-established bioassay platform that can accurately measure the target of MOA for various bispecific antibodies. For a lot of companies, they may have here because bioassay platform development is always time-consuming, and there are uncertainties in the assay development. However, in ProBio, because we have our own cell engineering group, it's much easier and for us to produce a robust bioassay platform. With this, I'm ending today's presentation, and I'm looking forward to the questions. Thank you a lot.
Thank you, Mr. Zhang, for the great presentation. So now let's get to the Q&A session. So we have received quite a number of questions for the audience already, and I'll read out to you to answer them, okay? So the first question, comparing with traditional monoclonal antibody, it is more difficult to develop a process for bispecific antibody. What's the time would you expect the timeline from DNA synthesis to batch release?
Oh, that's a very good question. So as the competitors goes hard, a lot of companies may really have the timeline concern when they're doing the CMC projects. So for bispecific antibodies, I would say for monoclonal antibody, the fastest we can do is 6 months from DNA synthesis to the tox batch deliver, because tox batch deliver is always in the critical timeline. And after the tox batch deliver, it's really depending on how the tox study goes, other than the CMC project progress. So if we want to apply the IND, get IND clearance as soon as possible, it's always important to have the tox study materials ready at the first place. For monoclonal antibody, it takes 6 months from DNA synthesis to the tox batch delivery. And for bispecific antibody, it's really depending on the formats. For the asymmetric formats, it takes a longer time, but for symmetric formats, it only takes 7 months from the DNA synthesis to the tox batch delivery.
All right, thank you, Mr. Zhang. So the next question, CHOK1 GenS cell line is relatively new to me. Would you mind to elaborate more on its productivity and also stability?
Yeah, that's also a very good question regarding cell line qualification. Our cell line have been on the market for more than 10 years. So the cell line have a clear and a traceable record from which is licensed from ECACC. On average, for monoclonal antibody projects, the average titer is more than 5.47 gram per liter, and 50% of the project got more than 5 gram per liter at the end of the process development. And for bispecific products, is really formats depending. For FC region containing bispecific antibodies, the titer is more than 7.5 gram per liter. And however, if the titer is a concern for the clients, we would recommend to do intensified fed batch by increase the inoculation concentration. We can double the titer with the same development time, and also without impacting the product qualities.
All right, thank you, Mr. Jiang. The next question from the audience is also related to cell line. So the question is, CHO-K1 GenS cell line is good at productivity, and the CHO-K1 ADCC 2B cell line is good at VDC. So can you tell us which cell line is better to produce the bispecific antibody?
Good question. So that's really depending on the molecule mechanism of actions. So if ADCC is considered the main mechanism of actions for this target product, we definitely recommend to use the ADCC enhanced cell line, which is also adapted from the CHO-K1 genes system. However, if ADCC is not considered a main MOA for this target product, we just go to the CHO-K1 genes cell line, which is a classic one in our company.
All right, thank you, Mr. Rubin. The next question. So based on your experience, what would be the main challenge for bispecific antibody CMC, and how would ProBio overcomes it?
So for now, for bispecific products, the main concern is still the impurity removal. And because a lot of times those impurities, they are similar to the target product. So we have to do resin selection, resin screening, and also do the really do the process parameters optimization in order to reach the optimal conditions. So there's no shortcut for developing a good process for downstream process development, especially for the bispecific product. So I would say we just go through more resin screening and to tweak the process parameters harder to get the best process for our clients.
All right, Mr. Jiang, thank you so much. So we have a few questions related to Dr. Walsh's presentation, so maybe we'll get to that first. So Dr. Walsh, I think this is related to your presentation. The image of GPC antibody staining was represented the difference between normal and cancer cells. Since tissue images are relative, it is difficult to prove that GPC antibodies do not attach to the normal cells at all. So can I check if you tried to conduct other analysis to confirm the off-target binding?
Yes, we have. So those have been animal imaging studies using both Cy5 labeled antibody as well as zirconium 89 labeled antibody. In both of these studies, we did not see any off-target binding in the animals. I agree that while it is IHC is relative, when you have a tissue that is expressing GPC1, particularly in aggressive cancer, it is very clear but the animal studies that we did certainly backed up the fact that it's very low, shall we say, undetectable levels of GPC1 expression in normal tissue. And certainly I think this is also backed up by the fact that we saw no drug-related adverse events in our safety study.
All right, thank you, Dr. Walsh, for the answers. So the next question from the audience, can I know how did you discover GPC1?
Yes, that's a very interesting story. So originally the parent antibody, which was called BLCA38, was invented by Professor Pamela Russell, who was a very famous researcher in Sydney at the Prince of Wales Hospital. Pam created this antibody by taking bladder cancer extract from a patient and making monoclonals in what you might call a shotgun method. So this was not purified protein to make the monoclonal, but rather a mixture. Pam then screened the potential clones with cancer cells using flow cytometry. So by definition, she was finding monoclonal antibodies that would bind to cell surface proteins. At this point, we became involved with Professor Russell and we in-licensed that original antibody. And we began to look for the antigen, GPC1. We found that it was very difficult to isolate, and we did not understand why until we found that it was actually reduction sensitive. So when we ran a Western blot, we would use reducing agent, we would lose the band. When we used non-reducing conditions, we were able to find the band, and this is because the epitope is a structural epitope rather than a sequential one. So it depends on the actual structure of the GPC1 molecule being intact. Along the way, we also discovered that the original antibody was in fact not a monoclonal, but it was actually two populations of cells, a biclonal. So we further cloned out our version of that antibody, which allowed us to have a new intellectual property around this. So we have a composition of matter patterned around that. But it actually took some years to find that GPC1 because of that sensitivity to reduction. Once we found what the molecule was, we were able to look in the literature to see that there had been some early studies on this molecule as part of the glypican family by two researchers in the US called Dr. Cork and Dr. Lander. And so we continued to look at potential cancers that they'd looked at as well as ones we were looking at. So that expanded the indications across not just prostate and bladder, which is where we had been looking, but also things like breast and pancreas.
All right, Mr. Jiang, Dr. Walsh, thank you so much for your sharing. So the next question, do you think that radioimmunotherapy will have a key role in cancer treatment in the future?
Yes, I do, and we are already seeing some radioimmunotherapies across cancer indications, and again, prostate is a good example. So some of the audience may be aware of the compound bsAb, and this compound, prostate-specific membrane antigen, so different to PSA, has been a tumour marker for prostate cancer for some time. They're now very successfully using a theranostic approach with a gallium-68 tracer on their PSMA molecule and a lutetium-177, but more recently and even more excitingly, using an alpha therapy, so using an actinium-225, they have been able to show complete removal of tumour metastases that have been throughout the body. So it's a very promising line of therapy for cancer patients. There, of course, been developments in other cancers as well, but I think the PSMA story is a very compelling one.
All right, thank you, Dr. Walsh. So I think the next question is quite an interesting one. So because since you are the founder for Glyterix, some of them are interested to know how did you become interested in this field and founded Glyterix?
So I began my life as a scientist. I always had an interest in medical research. I began researching in the field of allergy, and that interested me understanding what particular proteins caused allergy. From there, I began to research more mainstream immunology and inflammation, and this led me into the field of proteomics during the mid-90s. So I ran the world's first national proteomics facility housed in Sydney, funded by the Australian federal government. But during that time, I really found a frustration in being unable to really take the discoveries we made and turn that into intellectual property, which would eventually become products to benefit patients. So that inspired me to start my first company, which was in proteomics. But during the course of that, I began to work with Professor Russell and became interested in cancer biology and in this cancer therapy. So I founded the company on the basis of that. And we are very excited that we're now in the clinic and that we're moving to that phase 1B study. I think that is a fantastic place to be in, and we really think that the main part of this is seeing needs that patients don't have at the moment. They have unmet need in this area in cancer, and this may offer an additional therapy for them that will make a big difference in their life, has an opportunity to extend life. So it's really been a passion of mine for a long time to do that.
That's really inspiring to know. Thank you, Dr. Walsh, for the great sharing. So in your opinion, what are the key services that was needed by the startup companies to enable their success like yours?
You know, one of the key services is something that is applicable to many organizations, and that is for that partner organization to be able to deliver what they say within a reasonable timeline. We're all aware that there are sometimes external forces, supply chain issues arising from COVID and so forth. But what is key for a startup company is that your partner has good scientific understanding, capability to deliver in the kind of timelines that they say they can deliver in. And if they can do that, they can really not only help you reach your goals in terms of developing the product, but also in terms of the fundraising aspect, the capital needed to build a new product like this, because my investors are looking for me to hit my milestones. So I need partners who are able to help me do that. And of course, if we can also find partners that excel in their field, that are able to offer, the best service possible, that's fantastic. But, those key elements are really, really important.
Walsh, for the great sharing. It's really inspiring to know how you founded the company and how the company has success ever since. I think we have one last question, and I think this question is directed to Mr. Jiang from GenScript ProBio. So the last question from the floor, is there any recommendations for unstable product CMC? Mr. Jiang, maybe you can take this.
Oh, yes. So previously, I have been working in a company that's producing enzymes for those rare disease patients. So from that on, I have been working with quite a lot of unstable products. I would say for those unstable products, the right thing to do for upstream is probably perfusion process, because during the perfusion process, once the product is expressed outside the cell, then this product is quickly removed from the bioreactor into a stable, cooled tank that can make this product more stable. And for the purification process, of course, we have to consider the instability of this product, and you may have to do the purification process in cold temperature, or you have to do more intermediate stability study in order to support this unstable product to be manufactured future in the GMP environment. Yeah, that's all my recommendations. Thank you.
All right, thank you, Mr. Jiang. So I think we have covered all the questions from the audience, and that's all we have for today. So to all the audience, if you have any other questions, please contact us through our official email, and we will write back our answers from the speakers back to you. So thank you once again to Dr. Walsh and also Mr. Jiang for your time today to present such an inspiring presentations, and also to every one of you for attending for today's webinar. We look forward to meeting you again soon on our next webinar. Thank you and goodbye.
Thank you.
Thank you. Bye-bye.
Bye-bye.